Okay, so today is going to be probability and genetics. We're going to do about 3 or 4 pages all together. Today's date is April the 16th. Okay, 2 more days, woohoo, after today anyways, until spring break. We've done something similar to this before. This one is a little bit different in that the before statements are here and then the after statements are after the reading. So codominance of traits allows both the dominant and recessive trait to be expressed. This is something that's like, okay, there's a whole bunch of codominance of traits, allows for both the dominant and recessive traits to be expressed. Goodness gracious, who knows what that means? So maybe you don't have an answer. Maybe you're like, oh my gosh, I know all about this. I wrote a book about this, or I kind of have some idea. My answer, codominance. I'm going to put my question. So before reading the explanation, I don't really know. So what does codominance mean? Okay. Sometimes I'm like, I don't even know. I'm not too sure. But that's what I'll come back to. So I know that in my reading, or as I'm reading, I need to be thinking about codominance, dominant, recessive. I've got to come back for that one. Statement number 2, doctors could use a Punnett square to determine all the traits you have. Doctors could use a Punnett square to determine all the traits you have. I'm not too sure. A Punnett square? I thought doctors could use tests. But maybe you're really still not sure. I'm going to say more scientific tests. More scientific tests. Which makes sense. I mean, a Punnett square, how do they make a little box and fill it in? But so as you read, consider these questions. How does probability help explain Mendel's discovery? And then how do geneticists use Punnett squares? So Gregor Mendel was that monk. He was hanging out and he was doing the pea plants, looking at their different phenotypes of the purple and the white plants. And the reason he did this was you can visibly tell the differences between tall and short, and you can use the differences between. All right, so back to this because this took like a 10-minute break. Whoops. So Gregor Mendel, he worked with the pea plants. That's why he's so famous. He's called the father of genetics. So probability, anytime you see something in bold, probably important, is the likelihood that a particular event will occur. The principles of probability predict what is likely to occur, not necessarily what will occur. So they predict. For example, a coin toss. It should be a coin. The coin will land either heads, up, or tails. So it's a 50-50 chance. Each of these two events is equally likely to happen. In other words, there's a 1 in 2 chance that a tossed coin will land heads up. There's a 1 in 2 chance that it will land tails up. A 1 in 2 result can be expressed as a fraction, half, or as a percent, 50%. The result of one coin toss does not affect the results of the next coin toss. Each event is independent of each other. So just because it lands on heads 50 times out of 50 doesn't mean that it's necessarily going to be heads the next time. They're independent of each other. So each event is independent of each other. That's what it means. Just because it lands on heads one time doesn't mean it's automatically going to be on heads next, or it has some sort of influencing factor on it. When Gregor Mendel analyzed the results of a cross of his peas, he carefully counted all the offspring. Over time, he realized that he could apply the principles of probability to his crosses. Mendel was the first scientist to recognize that the principles of probability, so something you do in math, can be used to predict the results of genetic crosses. A tool that applies the laws of probability to genetics is a Punnett square. This will probably be, not probably, I think there's a couple questions on your NCFE at the end of the year that have to do with Punnett squares, which is why I'm making sure that we go over this. A Punnett square is a chart that shows the possible combinations of alleles that can be resulted from a genetic cross. Scientists that study genetics and heredity use Punnett squares to show all the possible outcomes of a genetic cross and to determine the probability of a particular outcome. In a Punnett square, all the possible alleles from one parent are written across the top. All the possible alleles from the other parent are written down the left side. The combined alleles in a box of the Punnett square represent all the possible combinations of offspring. And I'm going to get rid of this because we don't need this. Okay, so in a Punnett square, Okay, it's a little box. Mom's alleles or dad's, it doesn't matter whose goes where, and you always use the same letter. So usually B and B are pretty easy because you can tell the difference between a capital and a lowercase. So let's say this is mom, this is dad. Okay, doesn't matter which order you put them in. Big B, little b, and we have 2 little b's. If you use something like an S, it's going to be hard to tell the difference between capital and lowercase, so sometimes it's just easier like a big G, little g. But this could be mom or dad, doesn't matter which side. And then the Punnett square is going to show the probability of how the offspring would look based on the genetic types. So to use the terms that geneticists use to describe organisms are genotype and phenotype. Organism's phenotype is its physical appearance. How I remember that is they both start with physical. So a phenotype is what it looks like, or its visible traits. An organism's genotype is its genetic makeup or allele combinations. A genotype might not always be seen, so it might have a recessive trait, but it looks like the dominant trait. So I have my parents, my mom has blonde hair, blue eyes, my dad has brown hair, green eyes. I still have the traits from my dad, but they are not the phenotypes. That's not what's seen. When an organism has two identical alleles, and alleles in this case are just letters, okay, the organism is said to be homozygous. And when an organism that has two different alleles with the same trait is said to be heterozygous. Hetero simply means different, homo means same. Oh my gosh, you said homo, calm down. For all the traits in peas that Mendel studied, one allele was dominant while the others was recessive. This is not always the case. An inheritance pattern called codominance, so that goes back to our, I'm still using pen, that goes back to our question 1. The alleles are expressed, for example, in erminate chickens, the alleles for feather colors are codominant. Heterozygous chickens have both black and white feathers. So statement number 1, codominance of traits allows both the dominant and recessive traits to be expressed. That's true. So codominance, co just means kind of two. So strongly agree. It is now 3 o'clock, I think. Codominance, both alleles are expressed. Okay. Both alleles are expressed. All right. For question 2, after reading Punnett square, Dr. should use the Punnett square to determine the traits you have. It does say over here that geneticists use Punnett square, so that is going to be strongly agree. It's what geneticists use. It's what geneticists use. Cool. All right. So describe how the words heterozygous and homozygous describe similar and different traits. This one, homozygous, I'm just going to put homo and hetero because those are the two words that most likely stick out, homo and hetero. Homozygous just means describes when an organism has the same alleles. And to go back to the letter B, it could be big B, big B. It could be little b, little b. They're the same. Now, granted, they are the same letter, but in this case, if they're both capital or both lowercase, they're homo. And this describes when the organism has different, separate those two, different alleles. So in this case, it would just be big B, little b. And you always start off with your capital letter. Capital letter is always first. So it can never be little b, big B. It always has to be big, little. All right, because the big one means the dominant trait. That's the one that's showing. So when you're doing the genetic schemes, it could still have the recessive trait, but it's just not going to be visible. So it says describe the similarities between the word phenotype and genotype. This one's pretty easy. I'm going to kind of scroll up on this one a little bit. Pheno is just the physical appearance. Okay, so just the physical appearance. So what it actually looks like. Well, the genotype is genetic makeup. And it is, oh God, this stupid thing. Genetic makeup, and it is written in letters. Okay? They have to be the same letter. Could be one capital, one lowercase. So G, G, big G, little G, little G, little G. So homo, hetero, homo. All right? So hoes and he's. There you go.